When we talk about transport, we always think about the bustling cities, we think about cars moving, buses going, right? Or we think about taking public transport, getting from one place to another, making sure we're there on time. Or when we think about transport and movement, we also think about these beautiful animals going from one place to another. We go from one place to another, we locomote to different parts, and so do different animals. But at the same time, if you think about it, there are various microscopic organisms like amoeba that also have the ability that locomote and that transport to different parts. So if you think about it, transport of movement of objects, movement of organisms are seen across the world. And if you really think about it, movement is seen even in our cells. There are substances, there are molecules which need to get transported to the cells, out of the cells, within the cells, which is why today's class is going to be all about transport of molecules, that too especially across a certain part of the cell, which is the cell membrane. I'm your biology teacher Aishwarya and I welcome you to SHIKHAR series, wherein you guys are the future of tomorrow. And in the chapter Fundamental Unit of Life, we will exclusively cover this particular topic, that is transport of molecules that happen across the cell membrane. So I hope all of you here are excited for today's class. I hope that my audio, my video and my screen and what I'm writing on the screen is visible to all of you. If it is, please do give me a quick thumbs up in the chat to let me know that everything is good to go. Yes? And in the meanwhile, can we increase the font size a little bit more so that I can see things a little more clearly? Hello everybody, good evening, good evening. Now I know a lot of you here must be thinking, ma'am, when you say transport of molecules across the cell membrane, it says diffusion and osmosis, how hard could this be? But today we will not just be learning about diffusion and osmosis and what that is going to be, but rather we're going to understand that this cell membrane right here actually is very important because it allows for movement of select substances. And we will understand this concept a little bit more greatly, which means we will start off by exploring the concept of permeability. Now along with this, we will also understand how there are two types of transport. Now we see that there are two kinds of transport where we have active transport and we will understand passive transport. Now along with this, we will also understand that in passive transport is where we learn about diffusion and osmosis. But there's also active transport to understand. And along with this, when we understand osmosis, we need to understand that there are different types of solutions. And what would be the kind of osmosis that takes place? Is it an end osmosis? Is it exosmosis? So we are going to be dwelling a little more in greater detail than just learning blindly that there is something called diffusion, there is something called osmosis, right? So these are some things that we are going to be covering in greater detail. So I hope all of you here are excited for today's class. And the chat has been stuck on my screen, so we might have to refresh it once again. So everybody, please make sure that you have your notebooks and pens ready with you. We will also be having an activity room where we are going to be trying out something different, a different form of learning, where of course for all of you we will make sure that you just don't see. One thing is I can always give you some definitions and tell you this is what it is, right? But you know that if you've been a regular student of mine, you know that that's not how I teach you. And for new students who are very new in my class and you're wondering, okay, what really goes on in Aishwarya ma'am's class, you are in for an experience where, trust me, you will never forget these concepts. And once in your mind, I'm telling you, it's very easy, right? Very scoring part. And on an exam front, let me tell you, on an examination front, this topic will definitely 101%, I can bet my life on it that you will get questions. You will definitely get questions on this particular topic. So please make sure that you go ahead, hit the subscribe button on this video, because at the end of the day, let me tell you, BYJU'S 9th and 10th has got you covered. So are we all ready for an exciting class today? Yes? Are we ready? So quickly everybody, give me a quick Josh level check on the chat and then we're going to get started. Priya, I need to have more happiness, no sad faces here. Homework questions will also be there towards the end, so you have nothing to worry about. Yes? Okay, all right, amazing everybody. Let me quickly just set my hair and yes, let's get started with today's class. Now, quick recall from the previous class, we know that we started learning about the cell. We know how cell is the basic unit of life, basic structural and functional unit of life. And we know that there are basic components of the cell. That means whether I take a unicellular organism or I take a cell from a multicellular organism, right? So we know that some basic parts that it's definitely going to have is a cell membrane, is a cytoplasm, and it's going to be a nucleus. So now very quickly to refresh our understanding about cell membrane, right? So we know that the cell membrane has a fluid mosaic structure, right? So we know that it has different components. Now can you tell me what are all the components that make up the cell membrane in the live chat right now? And if you are watching this video much later, you can quickly refresh this in your head as well. What are the different things that make up the cell membrane or the structure of the cell membrane? So Gungun tells me lipids and proteins, but I need you to be more clear. Do we only say lipids? Yes? Very good. Shraddha says phospholipids. Exactly. So we know that the cell membrane is made up of phospholipids. Now, do we have a bilayer of phospholipids or do we have a unilayer of phospholipids? Single layer of phospholipids or do we have a bilayer of phospholipids? Very good, Saloni. So we have a phospholipid bilayer, which means that there is one like this and another layer here as well. So it has a phosphate head, right? And we know that it has a lipid tail. And along with this, we also know that there are various proteins also. Very good. All of you are telling me, ma'am, there are many proteins. Now, proteins are found on the outer side. Is it found only on the outer side? Is it found only on the inner side of the cell? Or is it found only across the membrane, which I'll call it as transmembrane? Or do we find all of these? Do we find outer, inner, transmembrane, or do we find all of this? Yes or no? Exactly, we find all of these, right? So we find some proteins which are present facing towards the outer side. We find some towards the inner side, some transmembrane proteins, right? And then of course we also have certain molecules of cholesterol as well, right? So we also see that there are certain molecules of cholesterol. So we see that this is the basic structure of the cell membrane. Now, of course, Ankita ma'am would have also told you that the function of the cell membrane is to basically act as a security guard in your school. Your security guard is only going to let if you have ID card and you are wearing uniform, right? Other than that, your security guard is not going to let you. So basically, the primary function of the cell membrane is to monitor what goes inside the cell and what goes outside. Along with that, it also gives the cell a certain shape and structure. So basically, when we say that things are going inside and outside, can I rephrase this and say that the cell membrane monitors transport, right? So we see that it monitors the transport of substances across the cell. Now, what substances get transported? Can you tell me what are substances which get transported across the cell? That means we know that cell is the basic unit of light. But what are the important substances which are getting transported? Study to success. I will answer your question in just a bit. Cell membrane is not made up of cellulose. Cell wall is made up of cellulose, which we will cover after this. Gases such as oxygen. Okay, very good. So we know oxygen is necessary for respiration to happen. So oxygen is there. Carbon dioxide, very good, Grisha, and many others. Yes, carbon dioxide is there. Now we also see water molecules are there, very good. So we see that there are water molecules which need to get transported. Now along with that, if you notice, we have some ions also. Let me point it out. We have some ions like your sodium ions, your potassium ions, you have your glucose molecules. So we see that there are various other substances also which require to be transported. Now thing is, membranes in general can be impermeable, it can be selectively permeable, and it can be freely permeable, right? So we know that there are these three kinds of concepts where we have impermeable membranes, selectively permeable, and we have freely permeable. How many of you remember what do we mean by impermeable? In the live chat, very quickly to recall this, please tell me what do we mean by impermeable? Yes, Vishnu, welcome, welcome. So I'm sure this is a concept which has been recalled by Ankita ma'am, but I'm doing a quick revision of this, right? Yes, very good. Does not. Impermeable. That means no substances are allowed to pass through, right? Which means that how much ever these substances come, it does not allow particles to pass through. While when I say selectively permeable, it means that there are select substances which are allowed to pass through it, right? Selected substances. Then we have freely permeable. Yes. So we have freely permeable, that is there, right? Freely permeable, which allows all particles to pass through it, right? Now many questions are coming. Ma'am, you are telling wrongly. Selectively permeable or semi-permeable? What is it? I know this question is there, which is why I am going to take this doubt. But before I take this doubt, right? I will take this doubt, then I will show you something. What is the difference between selectively permeable and semi-permeable? How many of you in the live chat right now have this doubt? Ma'am, you are using the word wrong. Selectively permeable and semi-permeable are the same thing. You should say cell membrane is semi-permeable. Why are you telling selectively permeable? Is there any difference at all, or is it more or less the same thing? How many of you have this doubt? See, if you have a doubt, that means it's good, right? It's good that you are thinking that way. Yes. Ma'am, is this class number 2? This is class number 3 of cell. Both are same. Okay, all right. Okay, so let me tell you one thing. Semi-permeable membrane is a membrane, or a semi-permeable membrane is that which allows movement of solvent molecules but does not allow movement of solute molecules. Okay. So this is what we mean by semi-permeable membrane. So if you think about it in your day-to-day life, right? So in your day-to-day life, when you think about it, for example, muslin cloth, right? Or if you think about it, there are these, potato sheet. There are these sheets which are available. Let me give you a simple example. How many of you have seen how your mama makes paneer at home, right? How does she make paneer? She will first curdle the milk, right? And then there will be some water as well which floats around. And at the end of the day, after the milk curdles, she will put one muslin cloth. She will pour it, right? She will pour that curdled mixture. There will be water that will come below, right? And all that protein that will settle, she'll squeeze it out like that. So what happens? We see that solvent molecules are going, right? She'll roll it in cloth. Exactly. So basically that is what we call as semi-permeable. If I have to give you a day-to-day example, allows movement of solvent molecules there, but does not allow movements of solute molecules. Now what is selectively permeable, right? Selectively permeable membranes, like our cell membrane, is normally semi-permeable, right? But in this case, selectively permeable allows for movement of solute molecules also, right? Which means our cell membrane, does it allow for movement of water molecules only? Yes or no? Does it allow for water molecules only? Yes or no? Cell membrane. Only water molecules? No. Because you know that some solute molecules, which is your ions, your glucose, your gases, they are all basically other molecules other than solvent, right? And they are able to pass through. So in this case, if you think about it, selectively permeable membranes are also semi-permeable. So if you say semi-permeable, it is not wrong. But if you say selectively permeable, it is more accurate. It means it allows for the movement of selective substances, which could be your water molecules also. So is plasma membrane semi-permeable or selectively permeable? Say selectively permeable, which includes your semi-permeability, right? So that is what we mean by this. Now, are we once and for all clear? What is selectively permeable and what is semi-permeable? Are we all clear? We are good to know, no? So tomorrow if somebody says selectively permeable or semi-permeable, oh ma'am, full confusion ho gaya. No? No, right? Cell wall is freely permeable. Exactly, it is, right? What is plasma membrane? Plasma membrane is your cell membrane, which is the outermost covering, right? Very good, very good. Okay, yes. Now, let's understand this concept of permeability. I actually wanted to show you one video before I get to this concept. So, I have a small video. Now when I tell you freely permeable, selectively permeable, how many of you are thinking, ma'am, I have to think so much to visualize it, right? How many of you are thinking, okay, ma'am, I'm visualizing it in my head, but I'm not able to really think about it properly. So how many of you are thinking this way, ma'am, if I could actually see what do you mean by selectively permeable or semi-permeable, I'll be able to understand it better, right? So many of you are there. Abir, thank you so much. Yes, okay. I can visualize, no problem. So what we're going to do is we're going to do a very simple, I'm going to show you a very simple experiment wherein we're going to run to my activity lab. We're going to watch it. Go to my activity lab, we'll see what we're up to and we will come back and discuss a little more. Yes? So let me quickly go to my activity lab. Hey there, welcome to my activity room where today we are going to learn about permeability of membranes by doing a very simple DIY. So without wasting any more time, let's get started. So as you can see, this setup is there right in front of me where we have a rectangular container. Now I have placed a membrane that is right in the center and there is water on both sides. So the first thing that we're going to do is label both of the sides. So on this side, I'm going to write A. And on the other side, I'm going to write B. So now I'm just going to move my A on top so that you can see this clearly. Now what we're going to do is I also have isabgol with me and I have chia seeds, things that we find in our kitchen. Now along with this, I also have OHP sheet that is there right here along with a scale. So what we're going to do first is we are going to take this OHP sheet and we're going to put it inside like this. So we're going to use OHP sheet as another type of membrane as well. Now once I place my OHP sheet, I'm going to take isabgol and I'm going to mix it and put it on side that I have marked as A. So let's take this and drop it and let's see what happens. So I'm going to use the scale to help me mix. So now we have mixed isabgol on side A. Let's give it a good stir and I'm going to hold it thoroughly. Now once I give it a good mix, you can see that isabgol is suspended on side A. But you don't see any of it on side B. Now let's see what happens if I remove this OHP sheet out of it. So I'm going to take it, it's a little messy but it's okay. And then let's give it a nice mix, right? So I'm going to mix it really well in such a way that we observe what's going to happen. So as you can see, I've given a good mix, maybe adding a little more of isabgol so that we can visualize what's going to really happen. Now if you notice, yes, with all the mixing that I have done, there's of course good amount of isabgol here. But on the other side, if you notice, when I removed the OHP sheet, you can see some particles of isabgol coming on side B. Now this is very, very important. We have zoomed in as well for you to get a good image. So on mixing, isabgol is able to travel towards side B. Super interesting. Now shall we make this even more interesting? Let's take our chia seeds that we have with us and we are going to put it on side B. Whoop, 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 whoop. All right then. So now I have mixed chia seeds, which are evidently bigger particles. So can we zoom in please so that they are able to see much clearly? So we're going to do a quick zoom in. Now you can see that isabgol is a little more whitish in color while chia seeds are the black seeds that you observe. Now as we keep it zoomed in and I mix this, right? We see that chia seeds are restricted only towards side B. We see that they're not really moving towards side A. Now what could be the reason behind this? It's very simple. The membrane that I have placed in the center, that is a semi-permeable membrane which allows the movement only of certain substances and it does not allow the movement of certain other substances. Well, there you go. Let's come back to my activity room that you saw me in right in the beginning. Right? So as you see with this very simple DIY, we learned about the permeability of membranes. The OHP sheet acted as an impermeable membrane which did not allow anything to pass through, while the membrane that I had placed right in the beginning acted as a semi-permeable membrane that allowed the movement of certain substances but not of all other substances. So with this, I hope the concept of permeability is clear to you. So how was that activity? See, it's always very simple, right? I mean, it's very easy for me to say, this is impermeable, learn the definition. This is selectively permeable, learn the definition, right? So in this particular case, I don't want you to focus on movement from high concentration to low concentration. No. Just that the first membrane, which was OHP sheet, was a solid transparent sheet, right? It was a solid transparent sheet. So we saw that there was no way in which the particles could move through it, which is why it was impermeable, right? It was impermeable. But the second membrane, we saw that had some pores like this, okay? So we saw that it was a mesh-like substance and we saw that there were some openings. But we saw that even the size of the particle mattered. Chia seeds that were there were bigger in size. So how much ever I mix it, it cannot go through it because it was bigger in size. But isabgol was small. So even the size of the particle matters, right? So when we talk about selectively permeable, there are many things that you need to keep in mind. Now, how many of you will never forget what is selectively permeable, semi-permeable and freely permeable? How many of you are never going to forget? Tell me in the class right now. Yes? Junior World and many others. 10th standard classes are going on in the channel. I'm going to take a 10th class after this as well. Yes? That is what I wanted, right? So now we know that there is all this kind of permeability that is there, but we know our beautiful cell membrane, right? Our beautiful little cell membrane is selectively permeable. But transport that happens across the cell membrane, right? That means to and fro movement, inside and outside and inside is of two kinds. Where we have transport, which can be active transport, and we have passive transport. Active means energy. That means energy is required for active transport. Passive transport, no energy required, right? So imagine just sitting and watching a YouTube video but not really dropping any likes, not hitting the subscribe button, just watching the video aise side pe. No energy, right? Very less energy. This is what we mean by passive transport. But when you are active like me, it means that active transport is there. Yes? So let's first understand passive transport. That means what are the different types of movement wherein we observe passive transport that does not require any energy. Now there are two ways in which passive transport happens, right? So that is diffusion and we have osmosis, which means the movement that we saw, kind of like going down the hill of a cycle. I mean, when we go down a slope, you don't really pedal, you just go and it moves, right? So we go from a place which is a high slope, right? Which means at a high point, it goes to a low point, right? And then you'll just sit like that from high to low, you will go. Pretty much similar concept that happens in diffusion and osmosis. So diffusion, let's understand this first. Now when I talk about diffusion, right? Observe this particular GIF right here. What do you observe? We saw that there was a drop of ink which was added, right? So we saw that there was a drop of ink which was added. Now it was added only in one spot where it was present in more amount. But we saw that there was relatively less at the bottom. So eventually what happened? We saw that it spread to different parts, right? So we saw that it spread to different parts. So in this case, diffusion can be simply defined as the free movement, right? So free movement of molecules. Yes, Kriti ma'am has already explained. Brilliant, right? So diffusion is nothing but the free movement of molecules from where to where? From high concentration to low concentration. Now, is any membrane involved? Do you see any membrane? Yes or no? Is membrane observed here? Yes or no? Do we observe membranes? That is what I need you to focus on. No, we do not observe any membrane. In this particular image, we don't see membrane. Next image, yes, membrane is there. But if you think about it, in the case of diffusion, there is actually no membrane. Even though across a cell, right, if you talk about a cell, we know that across the cell there are some molecules which will diffuse in and diffuse out. But actually, if you think about it, irrespective of whether the cell membrane is there or not there, it doesn't really matter. But we see that these molecules will go from a region of higher concentration to a region of lower concentration. And mainly we see that in the case of gases, right? Like in the case of gases, in some cases even liquids, we see, mainly gases I would say, we observe diffusion. So all your oxygen molecules, your carbon dioxide molecules, they all will be diffusing inside and outside. Ma'am, why does diffusion happen at the very first place? Very good question. Why is diffusion happening? Now, if you think about it, let's take a cell, right? Now, cell is doing so many activities. Cell requires energy, right? Cell needs energy. Now, for producing energy, oxygen is very important. Now, are we oxygen manufacturers that within our body only oxygen is produced? No, right? We are not oxygen manufacturers. Where is oxygen present? Oxygen is happily existing outside in the air. Happily chilling, right? Now, we need to make sure that oxygen is made available to our body. So, what do? We breathe in, right? We will breathe in and we will take all that air that is rich in oxygen. And this air will then, what will happen? Oxygen will enter into our blood. Blood will then transport it to different parts. And eventually what will happen is all this oxygen molecules is present in more amount outside. But inside the cell, do we have oxygen? Not that much. Maybe little bit here and there, right? Maybe because of some, we are not producing oxygen, but probably you can say there is no oxygen. So what has happened? There is a difference in concentration, right? So now we have observed a concentration difference. Now everybody will ask me, ma'am, what is concentration? You can equate it to amount. How much oxygen is present, right? So at this case, what will happen? There is difference in how much oxygen is present. There is more outside, or I can say concentration is high, and there is less inside. So eventually, because of this difference, oxygen will enter in freely, right? And this free movement is what we call as diffusion. Similarly, after it utilizes oxygen to produce energy, it will give out all this carbon dioxide. So much carbon dioxide it will give out. Now, if you think about it, outside the cell, very less carbon dioxide. Inside the cell, lots of carbon dioxide. Now, carbon dioxide, does our cell want to store and keep carbon dioxide? Yes or no? Does our body want to store carbon dioxide and keep it in nicely? Absolutely not, right? It's not good for the cell, which is why what's going to happen? Carbon dioxide will go again from inside the cell to outside. So this whole thing where there is free movement, right? We observe that there is free movement of substances. We call this as diffusion. Are we clear with the concept of diffusion? Now here it is always going to be from high to low. That is why we see that because the movement is from high to low, we see that there is no energy, right? I don't need to put effort. Aram se it is going to move, right? So that is what we mean by it. Now let's come to another thing. So we know that one kind of molecules or one set of molecules that have to be transported from our initial list was carbon dioxide and oxygen. Now we also know that water molecules have to be transported, right? So water molecules that is there also needs to be transported. So does water molecules also move from in the form of diffusion? Yes or no, right? I will take this question advanced bioscience once I finish teaching you that concept, right? Once I finish it. Now very quickly, I think a very important thing to target, right, is that till when will diffusion happen? How long is diffusion going to happen? Will it always be that from high to low it will keep coming? There is going to come a point when there is going to be a point where it stops. So when does diffusion stop? When will diffusion stop? Can you tell me? Yes? When is diffusion going to stop? When it becomes equal, exactly, right? I told you that diffusion is an example of passive transport. That means at the end of the day, only when the difference is high to low, you will have that. Kind of like going back to this very example, right? So if I go here, till I am at this point, from high to low when I am going, yes, movement is happening. Once I reach straight ground, right? Once I am on this plain road, do you think without pedaling I'll be able to go? No, right? I am going to be stuck there only. Done. My free movement is over. I am going to be stuck there. Which is why when we talk about diffusion, we see that diffusion is nothing but free movement of molecules from a region of high concentration to a region of low concentration until it is equal, right? Yes, when LHS is equal to RHS, that is what is going to happen, right? Now we saw one set of substances which has gotten transport, that is our gases. Now we also have solvent molecules or water molecules which need to be transported. So how exactly is water molecules going to get transported? To answer Ashu and many others question, ma'am, ye osmosis kya hai, right? Osmosis is what we understand as movement of solvent molecules. Now, how many of you have seen these? Can you tell me what is this? Can we please increase the comment font size? Yes? Prisha, I will explain this to you right now, but for that you need to understand this. Okay. Please tell me what this is. It is raisins. All right, very good. Everybody's like, ma'am, BYJU'S shorts pe we've seen this. Very good. These are kismis. Exactly. Now, kismis, you look at its appearance, right? Now, when you think about it, they all look wrinkled and saggy and they say, you know, fika fika. Now, after some time, they looked like this. How can I make my raisins go from this to this? Can any of you tell me? Yes? Yes, Harsimran has already given me the answer. Ma'am, if you soak it in water, then it will become like this. So when I soaked it in water, we saw that the raisins, which appeared to look something like that, went to looking to something like this. So how exactly has this happened? This has happened because of osmosis. So what is osmosis? Osmosis is a type of diffusion, you can say. But here we saw that there was free movement of molecules, right? Free movement of molecules in diffusion. But here we see that there is movement of solvent molecules or movement of water molecules, right? And where is water molecules going? Again, water molecules is going from a region of high concentration to low concentration. So I say, will it just go from a high to low? No, one very important clause is there. It has to be across a semi-permeable membrane, right? So here we can say selectively permeable, but again, like I told you, semi-permeable membrane allows for movement of solvent molecules. So when you're writing definition of osmosis, use semi-permeable membrane, right? So here if you think about it, what is osmosis? We have a membrane here, right? We have a membrane and here we see that there is movement of solvent molecules. Yes, so we see that there is movement of solvent molecules from a region from where to where? From a region of high concentration to a region of low concentration across a semi-permeable membrane, right? Because it's not that osmosis will happen only inside a cell. It can happen normally also. You can put a filter and osmosis can still take place, which is why you should say that semi-permeable membrane. But it is a phenomena you see in cells also. Are we clear so far? Are we all clear with what is osmosis? Nothing else. I know there are more concepts which are coming my way, but I want you to understand. So far, are you clear with me? So far, are you with me? Yes? Okay, done. Koi nahi baat karega abhi class pe, you pay attention. After that, you go talk in class. Okay? All right. Ashita, very good question which I will take in some time. Now, once again, I will explain. Osmosis can be simply defined as the movement of solvent molecules or movement of water molecules from a region of its high concentration to low concentration across a semi-permeable membrane, right? Now, what is the main difference between osmosis and diffusion? Now, I will give you some preliminary differences, okay? So, first and foremost, we see that osmosis is mainly seen in solvent molecules, while here normally we see this in gaseous molecules, right? But we also see it in other liquids and everything. Now, here mainly we see that a semi-permeable membrane is involved. Osmosis cannot take place without that. Semi-permeable membrane is a must. But here, semi-permeable membrane is not needed. So broadly we can see that these are two differences between osmosis and diffusion, okay? But when I talk about water molecules moving from or solvent molecules moving from high concentration to low concentration. Will they exist? Now let's imagine a cell. Okay, we are now within our body. Let's close our eyes, right? So let's close our eyes and let's imagine that we are inside our body. Okay, let's all close our eyes. We are all inside our body. We can see, right? We can visualize some cells. Just think about any cell you want to. It could be RBC, it could be neuron, or make it simple, make it skin cell, right? Let's make things easy for visualizing. Now think about it. You are within your cell. Okay, and now what are you seeing? You're seeing cell membrane. Okay, you can see solution. You can see cytoplasm. Everything is there. Now, when I talk about water or solvent inside our body, is it as water? Yes or no? Is it as water? Yes or no? Or do we find it as solutions? Yes? Do we find it as a solution, or do we find it as solvent separately or water as separately? Bimla tells ma'am, yes, it is not water, right? And many of you are saying, ma'am, no, it is in the form of solutions. So in this case, we see that we should recall solution concept. Now, for those of you who have come to my bridge class, this should be easy, right? But just for students who have not attended my bridge class and who are watching this for the first time, this is a recall for them. What is a solution? Solution is nothing but it is a mixture of solvent plus solute. Right? Solvent plus solute. This is what we mean as a solution. Now, based on how much of solute I add, right? Based on how much of solute I add, I can have different types of solutions. Yes? Are you all with me? Based on how much of solute I add, I can have more types of solutions. So based on that, we have three kinds of solutions where we have hypertonic, hypotonic and isotonic, right? So these are the three types of solution. But I have a question to ask. Now I am saying more solute, right? Or less solute, okay? I'm saying terms like more, less, same. Okay, I'm using these words, right? Based on how much of solute I add. Now, I have another question. Now, how am I saying more? More with respect to what? It's kind of like, I'll give you an example. If I say, I am short, right? I am short. So, for example, this is Aishwarya ma'am. I'm writing AR. I am short. Now, there is my friend Akanksha, right? Akanksha who is there. Then I have Ankita ma'am also. Now I am saying I am short. I am short with respect to who? If I go stand next to Ankita ma'am, I am short. But my friend Akanksha is shorter than me, right? Which means with respect to Akanksha, I am actually tall. But with respect to Ankita ma'am, I am short. So when I say more solute, or if I say less solute, it is not universal. It is with respect to something, right? So this term tonicity that comes, right? Tonicity that is there is basically relative difference, right? So this is what we understand as relative difference that is there. Yes? So let's understand this a little more. Now when you say isotonic, hypertonic, let's consider a cell, right? So this is going to be my cell. Yes? So a isotonic solution is a solution which has the same solute concentration as with respect to my cell here. Yes? With respect to cell concentration, exactly. Now I have hypo, right? Now hypo means less, okay? Which means that hypotonic solution is one which has less solute concentration, right? So we see that it has less solute. I'm going to put a down arrow. But if it has less solute, we see that it has more solvent. That means if I put very little salt in it, right? It has less solute in it. While hyper here, I'm going to use red color. When we say you are hyperactive, that means that you have more energy in you, right? So hyper means more. That means if there is more solute and less solvent, we call it hypertonic solution. Are you all with me so far? Yes? Are you all with me? Yes or no? Rough idea you got? Yes? Rough idea? Now, how many of you here believe, ma'am, if I visualize this, I'll understand this better? If you tell me more, if you tell me less, how will I know more and less? But if you show me more, if you show me less, I might be able to visualize it better. So how many of you want to visualize this? It's a very simple day-to-day activity. So shall we go to my activity room and see this once and for all, and let's understand this? Yes? And after that, we are going to learn more. Okay? So let's quickly go into our activity room and then let's come back to understand this better. So shall we go? Hey there, and welcome to my activity room. Today you and I are going to be learning about a concept that most of us get scared about, and that is about the different types of solutions. So we are going to use some daily objects that we find in and around our house to understand this super complicated concept, and we are going to make it so easy that you will never forget this concept after the end of this video. So without wasting any more time, let's jump right into it. So now, as you can see with me, there are 3 beakers here. I have one empty beaker in the front. I have a beaker with silver balls. I have one with gold foil balls. Now, let's start this particular session by first understanding what is a solution. Now, whenever I ask you what is a solution, what will you tell me? Ma'am, it is a mixture of solvent and solute. So let's assume that our gold foil balls are our solute and our silver foil balls are the solvent. So if I need to get a solution, I'm just going to mix these two together, right? And then I'm going to give it a good mix. And there you go. I have my solution with me. But now, of course, we see that there are different kinds of solutions, which is what scares most of us. So now let's quickly understand what are the 3 different kinds of solutions that we have. Now, one thing you all need to understand is that the 3 different kinds of solutions are the isotonic, hypertonic and hypotonic solutions. And this iso, hypo, hyper is all with respect to the tonicity. That means that it is the relative concentration. Now, what do I mean by the word relative? It is with respect to something. So, let's assume I have this red ball. And let's assume that this red ball is a cell. That means that inside the cell, there's of course cytoplasm, there will be some concentration of solute and solvent inside. So now when I say with relative to this cell, that means more solute, more solvent, all with respect to my cell that I will put in my solution. So I need you to keep this idea in mind when I use the terms hyper, hypo, iso. Yes? So let's go back to our activity room and understand this a little more in greater detail. So can we please zoom closer into the beaker so that our students are able to visualize? Yes, thank you. So now first, let's get started with something which is very, very simple, right? So let's get started with what do we mean by an isotonic solution. Iso means same. That means that if I take my cell, right, and I have maybe a certain solution with me. Now the concentration of this particular solution will be same as that of my cell. So we see that there will be no net movement of substances in this particular case. So this is what we understand as an isotonic solution. So I hope you are all here with me. Now let's get moving on to the next kind. So let me rearrange this a little bit and go to understanding something which we call as a hypotonic solution. Now hypo means less, right? So when I say hypo is less, that means that it has less number of solute molecules. That means there will be more solvent, less solute. And let me give it a good mix. Now how am I saying there is more solvent, less solute? Again, bringing back my one and only cell. That means my cell has more solute, less solvent. And my surrounding solution has less solute but more solvent, right? So as you all understand that, that is why I'm saying this is a hypotonic solution. Now let's move on to the last kind, which is hypertonic solution. Now hypertonic solution is something which is very interesting. So let me clear my beaker. Hyper means more, right? So in this case, it means that there is more solute, right? So there is a lot of solute, but very little, very little solvent. It will not have this much solvent. It'll have very little solvent. Then let's give us a good mix. Yes? So let's give this a good mix. And you see that there's more amount of solute, less solvent. When compared to my cell, which has more solvent, less solute. So this is what we mean by a hypertonic solution. So with this, of course, everybody, as you can see, with some simple day-to-day objects, if you know what is a solute and what is a solvent, the rest of it becomes very easy to understand. And I hope now the concept of hypertonic and isotonic solutions has become clear. All right, so now with this very simple, see, day-to-day things if you find in and around you, are you all able to understand this clearly? So quickly to rewind back, right? So we saw that there were gold foil balls which represented the solute. There were silver foil balls which represented the solvent, and we had a red ball wherein we saw that this was a cell. It was used as a reference. So when I say more solute or less solute, it is with respect to the cell, right? So this is what we understand by hypertonic, hypertonic. Yes? So iso, hypo, hyper, we are all clear? Are we clear so far? This more and less solute, are we all clear? Yes? Okay. Now let's understand what will happen. So I simply told you, I stopped at a very important point, that I have a solution, and if I keep my cell in it, right? So if you think about it, right? I kept my cell and I told you that this is the surrounding solution. But what will happen if I keep it in such a solution? That's what we need to identify, right? So most importantly, we should focus on this. Yes? So first and foremost, I am going to keep a cell, and I'm going to keep it in an isotonic solution. So what is an isotonic solution? An isotonic solution is a solution which has equal concentration of solute as the cell, right? So we see that if I say equal concentration, yes, that means amount of water molecules outside the cell and amount of water molecules inside the cell is more or less the same, right? So here we see that how much of a solute is also there is the same, how much solute inside is also the same. So if it is equal, will we observe any kind of movement? No, right? We see that in this case, we do not observe any kind of movement because it is the same amount. It is again, always remember, if you have to walk, right? If there is a straight road and if you are in your cycle, yes, do you think you will be able to cycle without putting any effort? No, right? You will be at the same point only. So at the same day, if I keep that cell in such an isotonic solution, it will remain the same size. Nothing is going to happen to it, right? So this is what we understand by it. But now, if I keep it in a hypotonic solution, now I told you that hypotonic solution has lower concentration of solute, which means if I have a look at the cell, now look at this second diagram here. This circle, I'm going to use, I'm going to color it, okay? Can you see this yellow color line, all of you, which I am coloring? What do you observe? Inside the cell, right? This right here is your cell. And inside the cell, we see that there is more solute, but there is very less solvent, right? Now, how am I saying there is less solvent? It is with respect to the outside solution, right? Because I told you your hypotonic solution is one which has lower concentration of solute. So what will happen at the end of the day? We see that more water is outside, less water inside. So is water going from outside to inside or inside to outside? Think about it. Water is more outside, right? Water is less inside. Why? Simply because there is more solute, right? Outside to inside or inside to outside? Very good, exactly. It will go from region of high concentration to low concentration. Go back to the initial example of our raisins, right? We saw those raisins or kishmish which we put in water. Now inside the raisins there was no water, but outside there was more water, right? Which is why from outside to inside it moved in. Now when it moved from outside to inside, what happened? It will swell, right? So we see that this will swell. And this kind of osmosis, this is one extra information for all of you, right? So this type of osmosis where there is movement of water from outside to inside, we call this type of osmosis as end osmosis. Yes? What is end osmosis? End osmosis is seen when we keep a cell in hypotonic solution where there is more solvent outside and less solvent inside, right? So because there is more outside, more water outside and less water inside, it will go from outside to inside. So we call it as end osmosis, right? Water is coming inside the cell through osmosis. Yes? Is end osmosis same as hypotonic solution? No. Hypotonic solution is the solution in which the cell is kept. End osmosis is the process by which the water moves in, right? So that is what we mean by this. Are we all clear? Are we all clear with this? Yes? Priya, I have explained it to you, bacha, once again. I hope it is clear to you now. Are you all with me? Yes or no? Don't worry about tonicity. Don't worry. That I'll teach in the end. Okay? I need this to be clear. Once again, we are keeping a cell which is kept in a hypotonic solution, which means it has more amount of solvent or more water with compared to the cell, right? Cell has less water, which is why water will move from outside to inside because it's going inside the cell by osmosis. If we see that, we call it as end osmosis. What is the opposite? Opposite happens in hypertonic solution. Now in hypertonic solution, we see that this particular solution has less solvent in it or less water in it, right? So the water is already very less. So if I compare my cell has more water, okay? My cell has more water than its surrounding. So inside my cell, lots of water. Outside, nothing is there, right? So now outside, because there is less water, what is going to happen? We see that movement of water or osmosis will take place from inside, right? So from within the cell, it will move outside, right? It will go outside. And we call this as exosmosis, yes? And what will happen? Because all the water is going out, it will shrink, right? So we normally see that in hypertonic solution, the cell will shrink. So hypo, cell will swell. But in hyper, we see that cell will shrink. Are we clear? Right? Are we clear? Now in osmosis, is osmosis happening in raisins or is diffusion happening? Osmosis is happening in the case of raisins, right? So that is what is happening. So are we all clear with this? Because this particular concept is very, very, very important. You will get application based questions. So now I am going to test you. Okay. So now I am going to test you. So your question is, a cell was kept in 15% salt solution for 12 hours. After 12 hours, it was observed that the cell shrank. Identify the solution and the process. Now identify the solution, right? What is this 15% salt solution? What type of solution is it? Is it a hypertonic solution? Is it a hypotonic solution? Or is it a isotonic solution? And observation is that the cell shrank in size. It shrank. So what would have happened? Exactly. So here we see that the solution is hypertonic. And what is the process that is taking place? Very good. It is exosmosis. Now in osmosis, can you tell me the direction of movement of water? Can you also tell me direction of movement of water? How is the direction moving? Inside to outside, outside to inside. Where is it going? I need everybody to answer. Ma'am, is there, Naman is asking me, ma'am, is it movement of water or solute molecules? It is movement of water molecules, okay? That is what we mean by that. Very good. Water is moving from inside the cell to the outside, which is why it is shrinking. Very good. So now, are we 100% clear? 100% pakka as to what is hypotonic, hypertonic, isotonic, right? We are clear with osmosis. We are clear with the concept. Very chotu sa concept, but very important concept, right? Now, last but not the least, we have active transport. Now, see, active transport that is going to be there. We see that in active transport, it requires energy. Now, why do you think it requires energy? Because in the case of active transport, it is normally seen in the case of certain ions, right? Or in the case of certain other molecules, right? Especially the solute molecules. So far we told water, gas and everything. But what about solute molecules, right? Solute also should move. So solute molecules pe what happens? We see that sometimes it goes from low concentration to high concentration also, right? It can be that possibility. So in these cases, we see that energy is required, where at times we see that in order to go from a region of low concentration to high concentration, ATP or energy is utilized so that you can move it. Think about it. If you have to climb up the slope, right? You are going from down, or I would say from low to high, right? And when you go from low to high, how much energy you need to put and cycle, right? So when you go up like that, lot of energy will go, yes? Which is why in this case, we see that in active transport, energy is required. So mainly in the case of ions, we see that this is necessary. Yes, Prisha, adenosine triphosphate is the full form of ATP. Exactly, mehnat lagta hai, of course. Ma'am, difference between exosmosis and osmosis. Arey, simple. Osmosis is nothing but movement of water molecules from high concentration to low. One type of osmosis is exosmosis, where here the direction is specified that it is from inside the cell to outside. It's as simple as that, okay? What will happen if we soak raisins in ink water? Actually, I have not tried. Why not try it? Why don't you all try it and let me know what happens? Yes? Why don't you all try soaking raisins in ink water and you share it with me on Telegram and you tell me what happens? Yes? Okay. So with this everybody, we are done, right? And quickly to recall. So quickly to summarize what I have taught you today. I mainly taught you about transport of substances across the cell membrane. Now I broadly told you that we have active transport which requires energy, right? And then we also saw passive transport which does not require energy or no energy. Yes? Now in passive transport, we learnt about diffusion, which we normally see in, oxygen, carbon dioxide, and then we learnt about osmosis, right? And then we also saw that there are different types of solutions, right? So here we learnt about the three different types which are there, which are isotonic, we have hypertonic, and we have hypotonic. And we also saw that osmosis could be of two types, where we saw that there is endosmosis and we have exosmosis. Yes? So everybody, with this ho gaya, right? Done. So here's a homework question for all of you which I want you to tell me in the comments of this video, right? Write the difference between exosmosis and endosmosis in the comments below, right? Now this particular topic is actually a very tricky concept to understand, which is why in this particular case, let me tell you that I hope by the end of today's class, you are clear with all the concepts that we discussed. I want to make it a point that these are very, very difficult concepts, but at a conceptual level, if you clarify all of your, doubts that you have, which I hope I've clarified most of your doubts, then trust me that it's going to be very simple and easy. And how many of you now feel confident about whatever you have studied so far? Yes? Are you all feeling confident? If so, you have to let me know in the comments of this video as well. Because you know, by the end of the day, BYJU'S 9th and 10th has always got you covered. And if you enjoy what we do, if you enjoyed today's class, let me know in the comments below. Do not forget to subscribe to the channel. For somebody who's asking me about why our fingers wrinkle, you know, there's a video on the YouTube channel also. You can check it out. But mainly we see that a lot of oil content gets out, and as a result we see that, you know, that's why you see those wrinkles. Yes? So everybody, I have a 10th grade class. My 10th grade students are already here telling me, ma'am, we have our class with you, so come soon. I am coming soon, so I will be signing off. I'll see you all soon. Up until then, bye everybody. Take care. Lots of love.